All right, thank you all for watching. I'm going to share with you some insights from work done in my lab at Colby College in Waterville, Maine in the United States. And we're interested in the biology of this insect here. This is the soapberry bug, Jadera haematoloma. And adults of this species can develop with either a long wing morph that you see at the top here, or a short wing morph like this one at the bottom. And the reason for this is that this species basically has a dispersal-fecundity trade-off. So a little more detail on the bugs. They're native to sort of the southern US and into Mexico and predominantly around the Gulf of Mexico. We're based up here, but we do a lot of field work down here in Florida. And these insects, you predominantly either see the flight-capable long wing morph here on the right, or this short wing form on the left. And these kind of intermediate individuals, they do occur, but they're fairly rare. And if we look more extensively across the animals, what we find is that long wing bugs have really robust flight muscles, like this one highlighted here, and short wing insects that can't fly are pretty much lacking that flight muscle entirely. Now, throughout my talk, I'll use these little cartoons to kind of indicate the long wing bugs that are able to fly and the short wing bugs here that just stand around. And from a lot of background research, what we know is that the determination for these bugs to either become long or short-winged is determined while they're juveniles. And if they experience a lot of food, not much competition for food, they tend to have the short wing form and they're not able to fly away and disperse, but they redirect resources towards higher fecundity. And individuals that get relatively less food as juveniles are much more prone to becoming long wing. They're capable of flight and they can disperse to locations where they might be able to find higher resources. All right, so getting into this system, I'm trained as a developmental geneticist, and so I was really interested to get a gene expression insight into the system. So we used a transcriptomic approach initially, and we started with full-length transcripts, and we produced what I think is a pretty high-quality transcriptome. There's almost certainly more transcripts than genes here, so ultimately we need to collapse these, but I think we've got most of the stuff here. And then in our later phases, what we did is we turned to 3' tag sequencing, where we can do many more samples and map those reads to the reference transcriptome or reference genome that we have, and really get a better sense of gene expression across multiple tissues, stages, etc. Getting into the system, we also wanted to produce a genome as a resource, and the first genomic insights were found in this species more than 100 years ago. A woman named Lila Porter at the University of Texas produced this lovely karyotype, and she identified 5 autosomes, an X chromosome in an X0 sex determination system, and a really interesting small heterochromatic M chromosome. This was something that she described as being precocious in anaphase and, at least to her description, not participating in recombination. So I found this really interesting. It reminded me a bit of the mammalian Y chromosome, where we might expect, because of low rates of recombination, for there to be lots of repeats, very few genes. All right. More recently, we had a colleague at the University of Texas who did a flow cytometry for us and estimated the genome size to be about 2 billion base pairs, so a fairly large insect genome. Undaunted by that, I had a postdoc join the lab, Devin O'Brien, shown here doing field work in Japan for his PhD. And one of the things he started off with was to inbreed Jadera for about 5 generations of full SIB matings, just to try to reduce heterozygosity in a genome of this size. And I think he was very successful in that. We sent some males off for genome sequencing using Hi-C assembly. And what we got back matches the karyotype very nicely. So we have 5 identifiable autosomes. We have what is presumably an X based on both its size and the fact that it gets about half as many reads mapping to it. And then we get a much smaller chromosome, but still much larger than the miscellaneous scaffolds, that we think is the M chromosome. So we're in the process of annotation with this genome right now, which is why it hasn't been published. But we have some annotation based on NTAP, which has been a really great piece of software to use. And looking at gene density across the genome, just using transcript start sites. So this is using our our transcriptome and just mapping using GMAP, I can already explode that first hypothesis. So rather than having fewer genes, the M chromosome appears to have about twice as much gene density as the other chromosomes. So really interesting. There's clearly a lot to explore here, like what might be going on the end of the third chromosome. Lots to follow up with. All right, so getting back to the transcriptome data, with this many samples, we had really strong batch effects, but DE-seq2 is awesome, and we can correct for those and get out clustering based on the tissues themselves, which is what we'd want to see. And then we can start looking at interesting factors like morph. And here we're using wing shape as a proxy for morph. This actually turns out to be a lot more sensitive. And our very strong expectation was that in long wing bugs, we'd see a lot of genes upregulated related to muscle development. And that's precisely what we see. We see things related to protein synthesis, respiration. And interestingly, we get a lot of signaling pathways coming out of this that are related to growth. We knew that FOXO and insulin was involved, and we have some confirmation here that things like TOR signaling, Hippo, FGF are also part of the system. Surprisingly, we saw very few genes that were differentially expressed in the gonads, but one of them that is this gene SOX14, which is known from fruit flies for a role in somatic follicle cell function. And so I'm really interested to follow up with this as a candidate explaining that difference in fecundity based on wing morph. All right, so related to this, there's another aspect of the biology here, and that is in this species, males also can be short-winged, and that's a pretty unusual situation for wing polyphenism in insects. And there's no obvious advantage to males being short-winged. In fact, in some populations, we actually see in crosses that short-winged males produce fewer offspring. And so that appears to be a really strong sexual conflict that's set up. And so we used our transcriptome data to try and get a handle on why that might be. And a couple of transcription factors jumped out as being upregulated both in long wing thorax and testis. And possibly this could relate to that male morph fertility difference. So these are mohawk and castor. And I was interested also in kind of looking again at the genome. And so many people will look for genomic regions of sex-biased gene expression. And you'd expect to see things like that on the sex chromosomes, but occasionally you also see things like that on an autosome. And so we just kind of use this method, scanning across the genome in a window, looking for morph-biased gene expression. And we can do this with expression data sets from any tissue we like. And we see some regions across the genome that have this bias and some interesting candidate genes in them, including that gene mohawk, which is, again, a really interesting candidate that may potentially explain the male reduced fertility in short-winged bugs, that sexual conflict that exists. So really interested to follow up on that. All right, so I just want to end and thank many of the people involved, especially the students who have worked on this project over the years, folks involved in the computation, sequencing, and especially Devin O'Brien, who helped out with the breeding for the sequencing. So thank you.